So today I'm going to talk about engineering multiscale health. And rather than just saying health, I want to really emphasize the idea of multiscale health. The idea here is, we've heard a lot about cell programming. How do you actually engineer specific cell states? But I think the thing that we're interested in is we don't care so much about the actual state of a cell, only what that means for the state of the organism. So actually, how do we figure out how to engineer the specific phenotypes we want at the organismal level? Okay, awesome. For some reason, the font looks weird down there, but let's just hope that's just a slide. Okay, so first, I want to just say, how do we actually learn from probably the greatest engineer of multiscale organismal health, and that's evolution. So you can imagine evolution has actually probably engineered almost any kind of trait that you can imagine in organisms. So even things that are technically immortal. So the immortal jellyfish can actually convert back into a polyp and then become another jellyfish. Bristlecone pines, extremely long negligible senescence. And then a bunch of other traits, regeneration or resilience traits. But the problem is evolution is not actually trying to engineer things that we want. So it's not going to actually engineer prolonged health or longevity unless it actually serves the purpose of what it's trying to do. So evolution is engineering systems for reproductive fitness. So this is kind of what the reward it's trying to achieve. And it can do this actually in very complex ways. So evolution doesn't understand the underlying mechanism of what gives rise to some of these traits. But similar to how you might think of a reinforcement learning model, it can actually kind of tinker until it gets the trait, and so it has a reward. It'll tinker, adjust a little bit, and kind of figure out how to get these traits that way. So one question is, can we actually change the reward system and do this on a much faster scale than what evolution would actually do? And probably the only way to do this is through something like AI. So AI excels at learning really complex rules, really complex patterns. And if you could actually build in silico models, you could actually ramp the speed up at which you could do these different experiments. And we know that AI has made huge gains in other types of domains that, like biology, are multiscale and highly complex. So hypothetically, we should be able to make huge gains applying AI to biology and learning from the other domains. And actually, this was already kind of discussed, so I won't spend too much time in it, but actually what has kind of machine learning, how has it evolved over time? So in the past, people used things like supervised machine learning just to build very specific types of models. But now we're kind of in this phase of foundation models that are being developed to actually have these kind of broad generalizable applications, and then you can fine-tune them or use things like reinforcement learning, potentially with human feedback to actually get them to be really sophisticated for whatever your application is. So even if we think about aging or biology, I think a lot of people are still kind of in this first phase. So they're building models for very specific applications and tasks. But how do we actually step back and build more kind of foundational models? I think we're calling things foundation models that maybe are not. So I don't know if just an RNA-seq foundation model is truly foundational. But actually, how do we build things that are actually foundational in biology? And I think when I talk to biologists, maybe this isn't as important for this group. They say, why do we want to build AI models? Mechanism's so important. I don't want anything black box that I don't understand. And actually, I think black box shouldn't be our enemy. It should be our friend. And actually, I would argue that mechanism could be the enemy in some case. So this is kind of a depiction of Pandora's box. And I think we spend too much time opening the box, trying to figure out what's in there. But again, we just want predictive models. I want to know when I perturb the system in some way, this is what I can predict the outcome would be. And that's really the only way that I think we're going to be able to accelerate to get to solve the types of problems that we want. Because biology, and especially aging, is so complex. I don't think a human is going to actually be able to understand the types of biology we're trying to solve for. So again, we're kind of stuck with this mechanism. It's kind of the tenet of biology, but really we need to move past that and just be okay with these predictive models. the biggest problem in biology, I think, and maybe this might be one reason why none of this will work, is we actually have a huge data problem in biology. So if you look at language, the amount of data you can scrape from the internet is enormous compared to the amount of biological data you can scrape from the internet. So we're really data poor in biology, and that's probably because each piece of biological data costs exorbitantly more than any piece of data in language. And it's not just that we don't have enough data, but I think the data we do have is actually problematic if we're talking about learning the landscape or language of biology. So humans are really biased. We tend to sample the same thing over and over and over again. So if you look across the commonly used cells, 60 to 70% of experiments are done in cancer cells. If you look even bigger, 80 to 90% of cells are immortalized. Do we actually think we're learning the language of biology just by looking at these cell types? You can look at these big data consortiums. So JUMP is all in U2OS cells. These are cancer cell line. Human Protein Atlas, their cell line resource. This is, I know you can't read the numbers. It's almost all cancer cells. Okay, so we can look in vivo. So almost everything is done in mice in vivo, and actually almost everything is done in a specific strain of mouse. So C57 Black 6, usually Black 6J mice. We know there's differences where most things are done in male mice. Most studies are done in young mice. Again, are we actually learning biology by just using the data that's available? And even if we think of modalities, so this is, you can't read the numbers again, but if I look on GEO, Gene Expression Omnibus, almost everything's RNA-seq. So if you think of omics, we're looking at the transcriptomics and very little else. And again, expecting this to capture all of biology. And I would argue that actually there's almost nothing multimodal that's truly paired out there. Oh shoot, 1 minute, okay. So actually, I think the key is actually do multimodal data. Because if you look at something through different lenses, you're actually going to get a more realistic picture of what it is. So to bring it back to the multiscale, what are we actually trying to do at Altos? We're trying to reverse disease and dysfunction at the organismal level. But the way we're doing this is actually intervening at the cellular level. And what this is going to actually require is a way to kind of bridge this multiscale problem. So you could have this kind of integrated discovery engine where you could actually predict if I did something at the cell level, what would that do at the organismal level based on the context of that intervention. And so we're actually building some databases that try and actually have multimodality and multiscale data that is truly paired and conditionally paired to bridge all the data resources we can also bring in publicly. And you, again, don't have to read this, but this is almost how you could depict data in hierarchical kind of biological fashion. So all of these are basically assays and readouts. These are different things you can represent with language metadata. And so to end with that, I'll just ask, do we actually have a data problem in biology? So this is actually the prompt for my workshop. So LLMs are really good already at solving biology, right? You can use agents and actually solve biological problems. So do we need more biological data, or has it learned enough by just looking across of all the compiled human knowledge in biology? I would argue that yes, we do, because right now, the information that's going in there is actually relying on the human's interpretation of that data. The human sees the data, gets some results, and all the LLMs get to see are the human's interpretation and results of that data for right now. So actually what I would pose as a challenge is how do we actually integrate the real biological data into the language models so that the models actually have the ability to pull out things humans would have never seen in the data, or maybe we're biased because we're trying to confirm some hypothesis, or we only look at things in very reductionistic ways. And with that, I want to do the most important slide, which is to thank Altos. So we have 3 institutes of science: Institute of Technology, Institute of Medicine. I'm in the Institute of Computation. Some of my great colleagues are also here, so if you want to ask any of them, can answer anything. This is not the entirety of the Institute of Computation. We have like 80 to 90 people, I think, but these are people who contributed to the thoughts that went on this slide, these slides. Thank you.
Awesome. Questions?
think you asked how are we gathering the data. So we're not gathering public multiscale data. We're generating data in-house. So even in the Institute of Computation, we have an entire group called the Data Collaboratory that's actually producing primary data for us. So again, most of this is going to have to be from mouse. We try and supplement this with human cells. And actually, we use diversity mouse too. But the key is, in one organism, how many kind of modalities at how many scales can we measure in that single organism? So can we measure different modalities for the cellular level, for the tissue level, for the organ level organism, and actually then be able to bridge, because you have things that are, I know these cells are in this tissue and I can say something about both of them and start to bridge things like that.